Good morning, everybody. I do speak up because pretty well coming from Stellenbosch University. It actually helps a little bit. I'm going to keep my fingers crossed that the slides has arrived here in one piece. I think I should just set my timer because I tend to ramble on and on. But it is a subject that I really, really enjoy talking about. Just a bit of background, I am a microbiologist by training. I'm actually a microbiologist by training. And my interest in soil microbiology comes from the fungi in soil. And it was purely an academic project. We were looking for penicillin species in famous sites. Because we had a hint that they were doing something in the world. After about 3 years of doing fungi in soil, we realized that there are bacterial communities and these things are talking to each other, and it's actually a lot more complex than we anticipated. So I thought today what I would do is just talk to you a little bit about the soil and the soil environment. Currently I'm working in the regenerative agricultural space. And the best technology that we at this point can afford to look at soil microorganisms. So I hope I can teach you something today, or at least I know I'm doing today a lot. So, when you speak to people, the first thing that they ask you is what is soil health? Yeah, I was just wondering if we could change my slides. Thank you so much. Awesome. And it actually all depends on who you speak to. I think the best definition that I currently can come up with is this one, which refers to soil health as soil quality, or the capacity of the soil to function. So when we talk about soil health, I would rather use the words functionality than soil health, because soil health is such a loaded term to use. Just coming back to soil, we have quite a bit of land, but just only 11 or 10, 15% of that is arable soil. And we all know working in agriculture that most of our soils are very low in organic matter. And that organic carbon tends to cause lots of problems because that is what our microbes need to live on in the soil. So we have a number of limitations. We have shallow soils, we have extreme textures with rocks, and we just spent a week at the Northern Cape last week speaking to farmers that actually farms in rocks. And very interesting, the first thing that they do when they prepare soils is going with the dynamite, which I thought was quite extreme. Seems to work. But the soil that they create in the process is just absolutely phenomenal to see. We do know that most of our soils are not managed very well, but there are an awareness now that we need to take care of the soil, we need to take care of the microbiomes in soil. Okay, so most of our farmers now know that yields are stabilizing, and just a side note, I'm not going to concentrate specifically on soy and Rhizobium, but there's going to be a lot of it in there. The profits has gone down. We did not know, we've read up a little bit, but 80% increase in costs in 2022 in terms of input costs, and 90% of our chemicals are important. Now, in the last week, that has come again. I thought about what this absolute fluctuation around dollar is doing to the cost of the farmer. It's a very volatile space to be in. So we have more and more farmers that go, but we do have this whole legion of organisms in our soils that can actually work for free. We just need to take care of them. We know there's about 10 trillion bacterial cells in every gram of soil. Please remember that number. 30% of the good soil consists of fungi, and they have numerous functions in soil. And many of these can fix nitrogen, and they can mineralize the nutrients for your plant. So they're critical in maintaining soil health. In regen agriculture, this is what we try to do, is to take care of that microbiome of the soil so that it can function and work. Regen agriculture makes the pillars are reduced with no tillage. No tillage actually helps that microbiome to stabilize over time. The use of cover crops, and I'll talk a bit about this later. We use composting, increase crop diversity, which increases microbial diversity. If I have time, I'm going to show you some of our results. Increased organic matter, use of biopesticides, biofertilizers, as well as integrated animals. Microbials, pretty interesting soil, have numerous functions. The more we learn about these organisms, the more functions we find. I just want to highlight the most important ones for me is decomposition in soil, decomposition of organic matter, and I heard a lot of my colleagues a while ago say that if it wasn't for fungi, we would be almost hip high in our own waste. So these things are crucial for getting rid of or decomposition. And then also the mineralization and uptake of nutrients. What is very clear is the more we know about, the more we learn about these organisms, the more we realize we actually don't know what they're doing. We have no idea what these things are doing and how they function. For a large number of them, they are unknown to us. We don't have cultures, we can't put them on plates, we can't study them in the lab. And all we see is that what they left for us, a little bit of DNA in the soil. So that's what we have. We make lots of inferences, but the picture is becoming clearer. What we do know is their function in soil, in terms of the microbes, not compartmentalized. These things work in consortia, so they never work on their own. They always have a number of others that work with them. It's actually quite noisy in soil. These things communicate with each other. They can excrete peptides. They talk to each other. They can shut other communications down. They produce all kinds of compounds to suppress growth or to increase growth. So it's a little bit noisy and it's a little bit like war in the soil. It's also not homogeneous. So what I collect in Florida is definitely not going to be what I collect in Stellenbosch. And depending on what your crop or your hectare looks like, it will depend how that microbiome looks like. Now for us, what we figured out is that the more homogeneous your crop, if you have monocrops, that microbiome is pretty stable across the lab. You can sample anywhere, you'll have a very similar microbiome. If you want to diversify that, bring in other plants. So things that influence your microorganisms, the biggest thing is pH, and the other one is water availability. So water availability can move your microbiome and completely shift it from one to a completely different one. And pH, and the other one is plants. And this is what we see time and time again, is that the plant that you plant have the biggest influence on the microbiome that it will attract. So we probably need to now dig into soil. So when we talk about the microbiome, we're really talking about the microorganisms that exist in the rhizosphere of your plant. That's where the function takes place. 10 centimeters away from your root, those organisms does not have a lot of influence on what is going on in the rhizosphere. So if you talk about the rhizosphere, there are a few regions. Of course, we have the area of cell division, the one that goes through the soil. It's got a little bit more habitat there for protection. It elongates just behind it, and then the third one up there, area about maturation. This works good. And this is actually really important. Work takes place from the microbiome. So this is a zone of microbial action. pH increase very incredibly important in this area. And most microorganisms like at around pH 5 is ideal for them to function, it's ideal for the uptake of nutrients. They can manage that, so the plant can manage that, but microorganisms, bacteria, in particular bacteria, can excrete acids to maintain that pH balance within the within that rhizosphere. So if you look at which bacteria and fungi are common there, so you have those that you guys know, the Rhizobium or the modulating bacteria. They tend to form symbiotic arrangements with roots, forming nodules, and these associations are pretty specific. And there's going to be other people talking a lot more about the biology of this. But then you also have fungi, AF, which is common to many of you. You've heard about the visceral mycorrhizal fungi, which has also evolved to follow this symbiotic relationship. And it's very interesting to know that this is the oldest form of fungi that has ever been found, is the visceral mycorrhizal fungi, and we found them in the roots of fossilized plants. So this is a very, very old symbiotic relationship. So I want you to think very, very small now. So if we talk about the rhizosphere, it's literally millimeters around the root. So if you really look into that rhizosphere, about 40%, 20 to 40% of whatever the plant makes to photosynthesis goes to your root as root exudates. And these root exudates can be sugars, it can be peptides, amino acids, alcohols, lots and lots of different molecules that is excreted into the environment or the rhizosphere, and they serve as number one molecules to attract, so attractants, alternatively as food sources for your microorganisms. Non-symbiotic bacteria in that rhizosphere can also modulate the immunity of the plant. So there's lots of functions within that rhizosphere. And if you have to guess how many, remember trillion cells per gram of soil, there's a couple of grams of soil around the root. You can imagine how many cells you have to your disposal to act as an egg. So how do things go in and out of the roots? So most of you should know this, I assume. We make usually lots of assumptions today. But most of the water, they just goes through osmosis. And we've had a lot of questions about how things go across and how bacteria enters and exits cells. So this is a bit of a big piece for me. Anything else, elements and nutrients go through active transport. And with this we mean there is an actual channel across that cell wall and membrane where the cell has to expend energy to get the nutrients across. So energy is really important for the plant. So it expends energy, so it has to make sure it's the correct thing coming over. Similarly, it uses active transport to get those root exudates out. So it is an energy expensive task for a plant. And these are the different molecules that would go across. So this is my attempt at being Pixar. So this is what you would kind of see in a very simplified picture of the rhizosphere. So you have the bacteria there, but they're floating around. For those of you who don't know, the charge on the root is negative, the charge on the bacterial cell is negative, so they tend not to be drawn together. So in order for them to get together, they really need to have communicate with the plant, and there are mechanisms in which they do that. And they do this through chemotaxis, which then makes some of our bacteria swim towards the plant and associate with them. We do this by, if we cut through a bacterial cell, most of our modulating bacteria looks like this. They have at the front of the cell, they have receptors. The receptors would recognize some of these signal molecules, which binds to the cell. It leads to a cascade of different genes being expressed in the cell, which kind of is just a trigger signal to tell the flagella to start rotating. It looks like a propeller on a ship. If you really go into the biology of these organisms, they're absolutely fantastic how they sense their environment. So we have a directional movement. They don't go straight like my little animation said. So what they do is they sense and then they'll go somewhere and then they will start sensing by spinning around. So it's a little bit like going through a bar when you're a bit drunk. We also have the vascular mycorrhizae here, and there's a very close relationship between vascular mycorrhizae and our modulating bacteria. Vascular mycorrhizae also depends on the plants for signals, and these are strigolactones. And strigolactones is a compound, a little peptide that the plant excretes to tell the fungus to start making those structures in the cell. So if you don't see that, it will simply not do that. So for those associations to take place, the plant has to tell the microbiome that they need to form that symbiotic relationship. And there are instances where they simply will not tell the cells to do that. So if you just step a little bit back, apart from these direct interactions, we have our modulating bacteria, the AMF, that is responsible for nitrogen fixation and solubilization of nutrients. We also have indirect mechanisms. And these could be antibiotics, so this is where you have non-symbiotic bacteria and fungi that acts as suppressors for other plant pathogens. And there are numerous ones of them. I think the most famous one that you guys know is trichoderma. We use it commonly as a biopesticide. We have by pesticide, and it shouldn't overcome, that's what it does. But it produces all kinds of other things. Mostly it induces the systemic resistance of the plants. So that's what it means. So there is a case to be made for maintaining a healthy microbiome. So apart from the nodulating bacteria, there's a whole group of other bacteria that can also affect atmospheric nitrogen. These are called free-living or diazotrophic bacteria. And these work in very close association, and I'm going to show you some of the stuff that we did with the nitrogen cycle, and they are very, very sensitive to the nitrogen levels in the soils. So just in case you forgot that I'm a taxonomist, currently these are the nodulating bacteria. They are not just one particular group, there are numerous different genera, they occur in different classes. So the ability to fix nitrogen is not fixed in one group, which I think is very interesting. And what is really important to remember is that plants secrete flavonoids that are very specific to certain Rhizobia or certain nodulating bacteria. So if it's not compatible, they simply would not combine, or they will not form that symbiotic relationship. So just to refresh your memory, and set the scene for today, nodule formation. It starts with the root hairs. There has to be a trigger for that to happen, and usually the trigger is low nitrogen levels in the soil. The low nitrogen levels in the soil triggers the plant to excrete the flavonoids, which then would recruit these bacteria if they are present, and that's a big question sometimes. So once they get to the cell, they've recognized those flavonoids, they move to the cells, they manage to attach to the root hairs, they would start forming, or the cell would, or the root hair would engulf them. And they do this through a very, very interesting cascade. So we have the flavonoids would go into, so it diffuse into your bacterial cell, attached to a complex called the NOT genes. The NOT genes get expressed as a protein. It forms a peptide that is called NOT factors that goes back into the plant cells, where it links into a NOT factor receptor on the cell wall, which then leads to the cascade for signaling this pathway to start. So this is really important. If those chemicals are not there and are not being excreted and are not recognized, nodule formation will not take place. And then, of course, we have the infection threat. So if everything works out and all of these things happen, then the infection threats will go into the cells, grows, and then the next thing happens. So the NOT genes, the NOT genes there, it's very, very light. It's now been expressed. When that happens, it triggers another cascade of genes in the bacterial cells. And it's called effector genes. And you'll see that my little bacterial cell has now formed a little infection tube into the plant cell. So we have the nodule, NOT factor receptors. We have the effectors that goes through this little infection tube into your plant cell. But also, your plant cell can recognize when your bacteria produces extracellular polysaccharides, which is kind of a gluey substance that it uses to stick to things. So most bacteria that can produce biofilms produce the substance, and your plant cell can recognize that. So now if you have all three of those things there, it would start inducing nodulation. This is the simplified form. What does happen sometimes, those effectors, if it's non-compatible host microbe, it will trigger a defense response. So that will prevent nodulation from happening. So it can also triggers that defense response. If it's compatible, the defense response is not triggered. And then hopefully, if everything goes well, you have nodules forming and you will be seeing some nitrogen fixated or fixed for your plant. I've noticed there are other people who will talk more about this, but there are various things in the soil that can prevent this. So high nitrogen levels, high salinity, high heavy metals, low phosphorus, and low molybdenum. Let's try this again. Molybdenum. I had it perfect this morning. Will prevent nodule formation. And what happens when you have those conditions, the stress of the plants goes down. As the stress goes up, root exudates goes down, and it recruits fewer bacteria. It likes a nice soil of pH of between 6 and 7. Moisture needs to be perfect. And in that case, you will have nodule inflation. And just to show you what the moisture does to your plant microbiome. So a couple of years ago, we looked at honeybush and rooibos, which is also a plant that forms nodules. They live in very, very nitrogen-poor soils, so they are absolutely dependent on these bacteria to fix their nitrogen for them. So what we did is we did high-throughput sequence analysis. So that's basically where we take all of the DNA in a soil sample, we sequence all of them, and we get a list of what's going on. So we can accurately determine which bacterial and fungal species are currently in the soil. So in this case, we only look at the bacteria. And when we compare bulk soils and rhizosphere soils, we couldn't see a big difference between them. So it's very similar communities. We thought that, commercial rooibos and native rooibos, maybe they would have different, but just look at the effect that the plant has. It doesn't matter where you plant that rooibos plant or how you cultivate it, it has its own microbiome. Also no difference between the farms, but if you look at the sampling times, we had a wet season and a dry season, how different those communities are. So we did network analysis, and basically this is just a co-occurrence network. So the larger the circle, the more abundant that group of organisms. And then you'll notice there are some faint lines in there. If it's a dotted line, it's a negative correlation. If it's a solid line, it's positive correlation. So this is during the wet season, highly abundant, a lot of co-occurrence, which is kind of for us an indication of interactions that takes place. And as soon as you go into your dry season, you'll see immediate change in how these organisms interact with each other. And you see some groups becomes really dominant. And in case of this one, it's the acidobacteria, which became very, very dominant. And they are absolutely adapted to grow in very dry, poor nutrient soils. Then remember, your rhizobia also competes with other organisms and native communities. You need some biscular mycorrhizae, which can enhance your nodulation. And just to show you what we mean by this whole thing, so remember we do photosynthesis. It takes CO2, it puts it into the root exudates. Root exudates is excreted, becomes food for your microbiome, and it can also get food from other sources. But when you start adding nitrogen to the soil, you break a very, very fine nitrogen limitation. So you have to have limited nitrogen in native systems to keep your carbon cycle kind of ticking over. If you break that limitation, you speed up your carbon cycle, which kind of changes things a little bit. What happens now is because there's so much nitrogen, your plants does not form those symbiotic relationships, and your microbiome becomes competitors. So they compete for the same nutrient sources. In general, not great. This is not what we do when we aim to go towards a regenerative or sustainable agriculture. You'll notice in the next one, the farmer's been nice enough to plant a cover crop there. Cover crops tend to increase diversity in the soils. Again, the same system. We have root exudates as food for the microbiome. Because we're not adding nitrogen, there's a little bit of a nitrogen limitation in the soil. It forces your nitrogen-fixing bacteria to start to fix nitrogen. They will then, of course, form symbiotic relationships with the plants. Mycorrhizae should also go in there if you have phosphorus limitation. Really, really important about mycorrhizae is that they are crucial for carbon sequestration. So they produce molecules which is not broken down easily by other microbiomes or other microorganisms, and they can sequester those carbon sources in deeper soil levels. So this is where we're going to. So when people ask you, do I have bad bacteria in my soil? We don't tend to tell them yes or no. Because I don't think there are bad bacteria and bad fungi. And neither are they good ones. They just are, and they survive in the environment that you create for them. The trick is how to manage that. So if you can measure those things, you can manage that environment. And to do that, you have to remember two laws. The one is Shelford's law of minimum or tolerance, which says that an organism has a minimum and maximum and optimum temperature where it can survive. That goes for temperature and pH and food sources and a whole lot of other things. So if you have a product that you're adding into an environment where those things are not optimal, your bacterium is not going to be functioning at optimal levels. The other one is Liebig's law of minimums that states that growth is dictated by the resource in the lowest or the limiting factor. Usually that is nitrogen. So it doesn't matter how much carbon you add to your soils, if you don't have enough nitrogen, you're not going to make any progress. So when we talk about these microbiomes, what you'd like to create within the microbiome is resilience. So you want to have a microbiome that can bounce back, that can protect your plants, and we see this in my diverse microbiomes that are fully functional. They are very resilient against things like drought, for instance. We like to build in some redundancy in there, and this we do with the cover crop so that you have a number of different bacteria living in the soil. So if you, for instance, have a drought experience and you have organisms that do better at drier conditions, they can replace them. But if they're not present, they cannot be recruited. So in agricultural practices, and I've used soybean pictures, I think these are soybeans. If you have a monocrop, you have a very soybean-specific microbiome associated with them. Some people like crop rotation, so they chop off their soybean, they leave those roots in the soil. No-till practices will leave those soils and the soil microbiomes intact. You can plant your maize on top of them. Maize, of course, will then recruit their own microbiome, which is very different to a soybean microbiome. It's not the same thing, but you have some overlaps. And as it grows, those microbiomes will develop. Some people go further and they add some crop rotations or intercropping, where you add additional plants there, and you can see that microbiome just keeps getting expanded and getting more and more diverse. And this builds into the functionality, and if you really want to go full regen on people, you add some animals to your system. And we've seen this in some of these systems. As soon as you add animals, it really pushes that system to a fully sustainable regenerative system. And what the animals does is, mostly, it fertilizes the environment. It brings new microbes in there, and very important to remember about those microbes. They put all organic nutrients in the soil, but they put in something called necromass. And in terms of the microorganisms soil, that necromass is so important. And it's just the name we give for dead bugs. And it contains a lot of nitrogen, carbon, and phosphorus. So it's a source of nutrients that we don't necessarily measure, but that are present. So it's a very recent paper. I don't think they published it yet, but I got this picture from that shows that as you increase your grazing, almost everything about your microbiome gets better. So I have 2 minutes to just show you a case study we did in the Free State on maize. So what we wanted to do is compare regenerative microbiomes with conventional microbiomes on maize. So we did it over 3 different sampling periods across the season. So this is across the season, fungi and bacteria, and we use the high throughput sequence method to do this. First thing you'll notice, the red ones at the bottom here is the bacterial diversity. It remains lower than the regen farms across the season. So less bacterial diversity. Fungi, not so much. And we think it's because fungi has such a defined function. It doesn't matter which fungi are present, as long as they can decompose, they're there, and they would dominate. So the stack bars here, these are just the different organisms, and if you look at the side there, this is the 50 most common ones, and it only represents about 60% of your microbiome. All of the other bacteria, so within something like this, you would have 1,000 to 2,000 species of bacteria. 50 of them form 60% of that microbiome. The rest of them, very low abundances, but they are there. And they form very important functions, even if they're there in low abundances. So size really doesn't matter always. Fungi, exactly the same, and what you will notice if you can read that list, which I assume you cannot, you'll notice there's a whole bunch of Fusariums in there, which immediately got the farmer all up in a tiff. Very interesting, he didn't have a lot of symptoms, and when we started really looking at those Fusariums, they turned out to be a number of different species. So the diversity in these Fusarium was much better, where in the conventional farm it was the single pathogen. So if you look at the how these things are put together, what they look like, the red one is conventional, the yellow is one of the farms. So the red and the yellow farm are literally divided by a road. The blue and the green farms, a couple of kilometers away. So where you plant makes a difference, but what you do makes much more of a difference. So we looked at the nitrogen cycle, and what we do is we use a method called qPCR to look at how many copies of a particular gene is present, which gives us an indication of how many organisms are present. So in the nitrogen cycle, we're very interested in this NIFH gene, which is very particular to nodulating bacteria and bacteria that can fix nitrogen. So they need that gene. So if we look at this across the seasons, conventional farms goes down, regen farms goes up. Okay, so sorry, that's me. Stop. Okay, I'm almost done. If you look at that particular gene, this is the gene that microbes use to convert ammonia into nitrate. It's a biological process. Look what happens. So the guy in the conventional farm has really done his full chemical fertilizer path. And they completely shut down that biological process. And I have to add, the other guys also do some chemical nitrogen. It's not like they're not doing anything. There is some cases, but they use it as needed. If we look at the nitrification, again, very low in our conventional farms, and it picks up. So this is the number of the bacteria that's present during those different times across the season. Then we also look at denitrification, which is also something we worry about. And again, you'll notice that it's quite high in your regen farms, which should raise a couple of eyebrows. But if you up your carbon, that is going to happen. But it is offset if you really look and go into the detail of these microbiomes. These farms also have a lot of methanogens that offset this denitrification. The same story here. And then, of course, another denitrification. Same picture. So, there is no ideal soil community. A plant, you put them in an environment, you create the environment, and they select what they need. And they do this based on their own requirements and the environment they live in. If you're trying to measure these things, please remember that the soil microbiology test should not be used as just a single measurement. It should be done in with a number of other tests, otherwise it doesn't mean a lot. Please remember your soils are very heterogeneous, so sampling matters. And soil diversity is sometimes a lot less important than soil functionality, which is when we do the diversity studies, we always link our functional test with that. So we look and see, if we have this microbiome, does it actually work? Because we have seen farms where they have the most beautiful microbiomes, highly diverse, highly redundant, highly non-functional, because you are just handing them the food. So if you just feed these things and you don't force them to feed themselves, they will be non-functional and become competitors. And with this, I'm going to leave you with a quote from a colleague of mine, said, soil health, like beauty, is in the eye of the beholder. And I would like to thank you for your attention. I'd be happy to ask some questions if you have them. Yes, sir.
Just remember to activate your speakers, please, for the red.
Hello?
Yes.
I don't have a question.
That was behind you. I get it a lot. It's because I'm wearing glasses. Let's try this one.
Good morning, everyone. I just want to ask two questions. The first one is based on the statement that low nitrogen levels enhance nodulation. Can you please quantify for me? Because where I come from, most farmers are pushing the yield, so they chemically apply nitrogen.
Yes.
So if you can tell me how much. And then the second question is, I primarily conduct research trials on soybeans, and what I saw this year was that I did not have any nodulation actually two trial sites. And I went to the weather data, And I saw from the 2nd of December until the 24th, temperatures were above 30. There was no rainfall, and there was very poor modulation. I even called NBFI and BASF people to come and check because it's an inoculation trial that was conducted on virgin soil. So my question is, I saw that you put the different things that also make modulation to be poor, and environmental conditions are not there. So I just want to know, do they also play a role, or was it just the soil and the rhizobium that was not maybe done the right way or stored properly, or what could have caused poor modulation?
Okay. Can I assume you inoculated?
Yes, the
Okay.
trial is a inoculation trial.
Okay.
So we do a control where we plant soybeans. Everything's not fertilized, and we plant them with the control that is untreated. And then I have different products, different names, but the bacteria is Rhizobium. But I have, like, for example, foundation pack, all those Nutrico and all of those products. But it's an inoculation trial.
Okay. So the first question is it's a difficult one, and I think there are some of my other colleagues may be able to answer that much better. What we found if you start looking at the microbiomes and not just chemical, I'm going to call it chemical farming, the measurement of the nitrogen in soil, the chemical measurement, does not actually tell you the big story. So people look at those nitrogen levels in the soil and they would either add to the soils, but what we don't measure is the biological nitrogen that's contained in our organic matter. So as soon as people start putting up their organic matter in soil, we don't measure that nitrogen. And I have a colleague in the US who's worked on, I think, maize. He's tried to look at it, but measuring that is incredibly difficult because the plant has an ability to, if you run us out of nitrogen, you will measure low nitrogen levels in the soil, but the plant has the ability to go and fetch that. If you have living microbes in the soil and they die off, they will provide nitrogen. One of the adaptations that the microbiomes have is to kill off half their populations for nitrogen. So they would literally kill off half the population of microbes to get nitrogen. So there's a lot of biological ways to get it, which we not measure. So it's really difficult to say if you start working with a biological organism and you use that chemical measurement to correlate that. But if you have high nitrogen levels, I can tell you then probably this modulation is not going to happen. But I don't know how low should low be. The next question was the temperature. What was the temperature at the roots?
It was anything between 30 to 36.
Yeah.
And that was and it was dry. And
so you probably now putting organisms into a system that's not optimal for them. So they do need the water. So you remember I showed you that they swim. They cannot swim if they don't have water available. So if there's no water at the roots, they cannot get to the roots. That's the way to move around, is through water. So that could be one problem. The other is the temperature. The temperature is kind of at the upper part of where these organisms function. They're okay-ish at 30 degrees. When it gets to 34, 35, they start to struggle. They really like a nice balmy 26 to 28. But we don't have those conditions in the soil, so which is why I'm asking you, below soil, usually it's a little bit cooler if you have coverage over your soils. So and that's the other idea that people have with the cover crops, is to reduce the temperature in soils and to create that cover that would prevent moisture from evaporating. So I think that there could be a number of answers to that question. One is suboptimal conditions. It's very dry. They don't like the lack of water. It's one of the big drivers of the microorganisms is water. But yeah, I hope that answers half your question or a quarter of your question. Yes, sir.
Good morning, Prof. I'm also Afrikaans, so I'll try to use my English before it runs out.
Jy kan maar Afrikaans praat, dis reg.
Let me try English, I think. So we work with farmers. If you think about the pressures that they starting to get, whether it's regulations or climate variability, the conversation of conservation agriculture, regenerative agriculture is quickly becoming an important topic. And soil health, we try to understand it and be able to guide our farming communities on that. So if I were to listen to your story from this morning, the type of plant that the farmer puts into the ground, first of all, we talk about exudates that it releases that attracts certain types of microbes. So on that thought, there's two questions. Is the microbes already in the soil, or is it brought in through the seeds? Okay. And I guess the follow-up question is, when we talk about now the diversity and the functionality of that microbial community in the soil, then the follow-up question is, the final question is, what can we do to ensure that the functionality of that soil community is optimal?
Yeah. So those are two very, very big questions. So to answer your first question, you don't get soils without microorganisms. And if you look at the recruitment of, if you think about 2,000 species in the rhizosphere of bacteria, the majority of those are present. And I've read a study a couple of days ago. This is really awesome research where they looked at the recruitment of these organisms. So as you move, and they started looking at from 5 millimeter to the rhizoplane, so just that bit, in that little bit of area, they start recruiting there, and it's just generalists. And as it goes towards straight onto the root, it becomes very specialized. So they look into this group and they recruit. Now the specialists like Rhizobium and AMF, when we looked at virgin soils, sometimes they're just not there. And we do analysis for farmers on a fairly routine basis, and we can talk about the analysis if you want to. And sometimes, that is the problem. They send us soils and say, do I have rhizobia? And we literally sequence everything to find those rhizobia. So we don't even try and plate them out. We sequence everything. And mostly when they are present in very, very low abundances. So they're not massively big colonies of these things. So it's really important then to make sure that the strain that you have is compatible with your plant and that the conditions are optimal for the plant to be able to send the signal so that the rhizobium can connect. And also remember, these things cannot swim for a meter. They small distances, so they need to be as close as possible to the root in order for that to take place, because those signal molecules work through diffusion. You need water for that. So if it's a dry environment, you're going to struggle to recruit. That area of recruitment becomes really, really small. And it really limits the chance of finding the correct one. In terms of inoculation, usually in the Free State, we've seen that if your neighbor does conventional agriculture, they will inoculate your fields over his fallow season. So your inoculums come into wind, seeds, composting, the organic matter that you have on your fields, your cover crops, but it can recruit that. So think about, if you look at the bulk soils, there's the sea of organisms. If you plant your plant, it recruits a section of them. Okay. And it's not that species specific for the majority of the microbiome, but it tends to be quite genus and class specific. But it is spread across the bacterial kingdom. Does it make sense? Awesome.
question, Karin?
Yes.
If I can get the right language. Regarding the flavonoids, you
Yes.
mentioned that the plant exudes them. Do we have any idea of the specific flavonoids that are associated with the specific crop? And could we then artificially apply those flavonoids to induce the process? Because I'm thinking if you have
That's a good question.
supplemented nitrogen, but you're not getting nodulation because there's too much nitrogen, but you apply the specific flavonoids or polyphenols, then perhaps you can induce that process.
I think you need to ask the people who really work with rhizobia and flavonoids. Yeah, so I just assumed you got this. So that question will be answered later. My gut feeling is it's going to be tricky. If there are no more questions, thank you very much for your attention.